Okay, so let's finish up our cancer section here of this cell division lecture. Just to sort of finish off here with these diagrams for clinical trials. What's interesting about it is the following. You can see that especially the green bars going up over the years. So up here is by year 2014, 15, 16, 17, 18. And the green bars are targeted drug therapies, new ones that are in clinical studies right now. And that's very encouraging because these are very, they have lower side effects and they really target specific type of cancer, and many of them have really good success. Down here, there's a projection again. So blue are new hormone type of drugs. In red, those are chemotherapy type of drugs, more traditional type of cancer drugs. And then the new targeted drug therapies, these are in green. That's very encouraging. So projected for 2025 that new clinical trials, it will go up, and that should really help many people. So here's something interesting too. I mean, at first sight, this table might not seem interesting to you at all, but the point here is that for every cancer, you can take a look at where's the high incidence population, where's the low incidence population. The high incidence population on this side and low incidence population on that side. And the difference is pretty extreme in many cases. So let's just give an example. Let's take colon cancer. So here we have colon cancer, high incidence population, and this is the incidence per 100,000 of the population. 34 for the United States, and the low incidence population over here, they're in India, and it's 1.8. So why is that? It's all humans. How come there's such a big difference? Or let's do first one, lung cancer. High incidence population here in the United States, 110 per 100,000 of the population. Low incidence population here, only 5.8 cases per 100,000 of the population. So a big difference. The question is, why is there such a big difference? And you can go across the entire chart and look at it and find other examples. But so why is that? And the obvious place to look first would be your diet. And so many years ago, by now, there was an initial study that was looking at the effect of your diet and lifestyle choices on the likelihood of developing certain cancers. And this is just one study of many. There have been summary studies now out there, and the World Health Organization published a couple of years ago, they published sort of a summary study that then concluded that processed meats are really bad for you, and they're now in the same category as cigarette smoking. So there's a pretty big difference here. But let's take a look at this. So here in this chart, what you can see is that particular study found certain lifestyle choices are beneficial for you. The higher the green bar is, the better it is for you. And big surprise, fruits and vegetables are really good for you. I'm sure you've never heard that, but here it is, brand new information. Eat your fruits and veggies. And you can lower your risk of all of these cancers right here. For example, colorectal cancer, really up high there. And a couple other things to point out. So physical activity is good, especially to avoid colorectal cancer. Refrigeration, so that you don't eat a lot of spoiled food, is really good so that you can avoid stomach cancer. And you can take a look at this chart, but overall, I don't think there's much of a surprise here to you. And on the negative side, let's take a look at that too. Here we have alcohol consumption. It's not good for your liver. And then whatever, especially high-proof alcohol, when it comes in contact with membranes, it's not good. So in your mouth, larynx, esophagus. So that mostly the high-proof stuff. But in general, and when it comes to alcohol, it's really mostly about moderation and avoiding the high-proof stuff, so the vodka and those kind of things. But overall, you are increasing your risk of developing certain cancers with alcohol consumption. And there are many other things that are not so good. Some things came as a surprise. Some people were thinking that certain things are not good for you, like coffee, that turned out not to be true. And you can take a look at the list right here. Obesity has been found to be a risk for developing endometrial cancer, also seems to increase your risk for breast cancer. And then here, the big bad guy off to the side. The bars would be so high that it wouldn't fit the chart. So they made it off to the side. Smoking tobacco is really, really, really bad for you. And again, this is probably coming as a huge surprise to you. So let it sink in and think about it. Do not smoke. Don't vape either. Those are things that are not good for you. It increases your risk not just for obvious types of cancer such as lung cancer, but then also wherever the cigarette smoke comes in direct contact with, in your mouth or nasopharynx area, but then also pancreatic cancer. And this is a cancer you really don't want to have. That one has a very high mortality rate. It's really difficult to treat. And then cervical cancer, bladder cancer. I'm not sure how the connection is with cigarette smoking and bladder cancer, but apparently it goes up really high as well. And here, one final slide here. A word of warning. If you are doing several bad things that increase your risk for developing certain cancers, then they may have a synergistic effect. That means they don't just add up their bad effect, but they multiply it. And this is true if you are drinking a lot of alcohol and you're smoking cigarettes. So here is the risk for non-smokers, but they are drinking various amounts of alcohol. Here is the risk for cigarette smokers, and then here is the risk if you are smoking more than 2 packs of cigarettes per day and you're drinking more than 4 alcoholic beverages or have more than 4 alcoholic drinks per day. This is where you're going to be, and that's really not where you want to be. So the risk is astronomical right there. Not a good lifestyle choice. So this concludes our discussion on cancer. There's lots of information out there on the internet. I'm sure you've seen something too. And we're going to move on now, though, to our last portion of this lecture, and that will be meiosis, which is known as the reduction division. So meiosis is a special type of cell division that only happens in gonads and is only there to produce what's called gametes. So we're going to have to take a look at the human life cycle here. We all started out as a zygote, so the product of the fusion of 2 gametes, an egg and a sperm, and that brought the 2 sets of genetic information from mother and father together and created a whole new individual that is diploid. That means you have inherited a set of genetic information from your mother and one from your father. So sets of genetic information are denoted by N. So 1N plus another N makes 2N right here. And that means we have 2 times 23 chromosomes. So 2N equal 46. And that is all of our regular cells. They have 46 chromosomes. But a single set is 23. And I'm sure you have heard of these sequencing sites. They put up give you information about your genome, and 23andMe is one of them. 23 because that's the single set of chromosomes, so N equal 23 for humans. So once that zygote has been created, then from there on out it's just mitosis, just copying what's there until this individual turns into an adult, and then it's time to do meiosis, which is the type of cell division that creates cells that are 1N again. Remember, all of our regular body cells are 2N, 46. And these gametes, eggs and sperm cells in the human case, they need to be 1N. So let's take a look at a human karyotype. That's this thing right here. So if you were to list all 23 pairs of human chromosomes, then you can roughly sort them out by size. We have 22 pairs that are called autosomes, and then the last pair, the 23rd pair, those are the sex chromosomes. Now, if you have 2 X chromosomes, then you're female, and if you have an X and a Y chromosome right here, then you are male. So this is the karyotype of a male. And here, quick comparison, mitosis, meiosis, recap from mitosis portion of the lecture. Over here, we are 2N to start with, and we're 2N to finish. The only difference is that when we start mitosis, each chromosome has 2 sister chromatids, so 2 arms. You see the red one has 2 arms, the yellow, the green, the blue, they all have 2 arms. And when they're down here after mitosis, they're still diploid. They're still, all 4 chromosomes are still there. It's just that they don't have a sister chromatid. They have only a single arm. You see that? Now what happens in meiosis is that we're going to split the pairs apart. So we need to line up the pairs. First the pairs have to find each other, and then we need to line up the pairs. So here we go. The 2 big ones right here, the blue and the green, that's a pair. They have to find each other. The little one right here, the smaller pair, the yellow and the red one. And the different colors just they denote that these came originally from your 2 parents, so 1 from each parent. And once the pairs have found each other, then you can do what's called the reduction division, where you're separating the pairs, and after that first step, the cells are haploid. They're no longer diploid. So that's the big difference right here. So that would be a meiosis 1. And after that first step, see, note how the remaining chromosomes, they still have 2 sister chromatids. See, the green one has 2 arms right there, and the yellow one too. But you don't have the blue one at all anymore. So these cells are now 1N. They are haploid. They only have a single one of each chromosome. And then the second part of meiosis is just like a regular mitosis, where you're splitting the sister chromatids apart. So down here you can see each one of these resulting cells. The chromosomes that are there, they only have 1 sister chromatid, only 1 arm to each. But the result of meiosis is always 4 cells, and they are haploid. Okay, so let's take another look at this here. So meiosis is known as the reduction division. It's the type of cell division that occurs in ovaries and testes, i.e., your gonads, if you don't want to specify male versus female. And the result of meiosis, the production of gametes, if you want to specify, that would be ova and sperm cells. There are 2 sequential divisions. The first division is the actual reduction division, where you are making haploid cells out of diploid ones. And for that, the homologous chromosomes, these pairs of chromosomes, they have to find each other and line up side by side, and then we have to pull the pairs apart. The result is that you're going to have haploid cells. So that would be, each daughter cell contains 23 different chromosomes for a haploid set. However, each one still has 2 sister chromatids, and that's what we're going to take care of in the second division. In the second division, each daughter cell divides with duplicate chromosomes going to each new daughter cell. So those are identical copies. So each sister chromatid is an identical copy of the other. It was done in S phase of the cell cycle. And the result is always 4 haploid cells. When we do spermatogenesis in the testes of males, we're producing 4 equally viable sperm cells. In oogenesis, in the ovaries, female ovaries, we're going to produce 1 extra large maturing egg cell, and the remaining 3 are polar bodies, and they will die. So the general theme here, without considering oogenesis versus spermatogenesis, the general theme here is listed. So you have 2 sequential divisions. So we have meiosis 1, that's until here. We're going through the same phases: prophase, metaphase, anaphase, telophase. But we have here prophase 1, metaphase 1, anaphase 1, telophase 1. The result would be 2 haploid cells right here. So that's the result, 2 haploid cells. They go straight into meiosis 2, and then we have this full sequence again: prophase, metaphase, anaphase, telophase 2 attached now. Okay. So, and the result will be 4 haploid cells on the bottom right here.